Thank you for viewing this video. I'm Emi Kaneko from the Japan Science and Technology Agency, JST. I'm director of JST's Singapore office and the owner of JST E-Pavilion. We are pleased and honored to be a key partner of Tech Innovation 2021. For this year's Tech Innovation, we present 9 SMEs and research institutions from Japan who will exhibit their technology solutions for the healthcare, food, and manufacturing industries. The wide variety of solutions include high-intensity focused ultrasound treatment for cancer, novel method of T-cell therapy for cancer, powered prosthetic legs, whole body sensing technology, pursuing algae's potential for sustainable food supply, and more. As a government-affiliated funding agency, we at JST are very much focused on solving complex social challenges through research and development. To bring the result of research to society, we support academia-industry collaboration and technology transfer, and have special programs to convert high-potential technology shifts into market-ready products. The COVID crisis has reminded us that we need global-scale cooperation to tackle global-scale problems. That's why at JST's Singapore office, we are actively promoting science and technology cooperation between Japan, Southeast Asia, and the world at large. We deeply value our association with IPI, the organizer of Tech Innovation, and believe that this kind of platform is essential to build connections and discover new opportunities for mutually beneficial cooperation in the region. Though we're presenting 9 Japanese technology solutions today, there are high potential sheets being developed all across Japan that are keen to cooperate with international partners. If you're looking for a particular technology solution, please don't hesitate to contact us at the JST Singapore office, and we'll be very happy to help connect you to a future partner. Thanks again for watching this video, and I hope you'll find it inspiring. Thank you.
Cultivating algae's potential for a better future. Arigarebayo is a clean tech startup from Japan, and we strive to solve global issues and contribute to the future of people and Mother Earth. If this rings your bell, we would welcome you to join our journey. My name is Amane Kimura, and I'm CEO of Arigarebayo. Let me start with what we are about. Arigarebayo is a clean tech startup that is established based on 20+ years of solid scientific research at the University of Tokyo. We aim to partner with industrial leaders to pave the way forward by delivering algae-based sustainable solutions that will empower many industries. And our purpose is to address not just the needs of society, such as depleting food and natural resources due to the growing population, but also we want to address the world's major concerns, such as global warming. More specifically, these are the issues we are addressing by our algae-based clean technology. First and foremost, wellness. As our expected lifetime has been extending, often referred as the 100-year life, wellness or well-being has become one of the key metrics to our lives. In the meantime, countries like ours, Japan, has been facing challenging dilemma with the social security system due to aging population. Secondly, food crisis. I strongly believe that our conventional food supply chain is not sustainable enough to feed increasing population with changing diets. It will be crucial to provide more sustainable options to this industry. Lastly, but not least, algae-based clean technology could play an important role in circular economy too. For instance, one of our projects has shown an interesting results for algae-based biodegradable plastics. Thus, our playing field is very broad, But next question you might be asking us would be, what can you do? What is your uniqueness? So this photo represents our uniqueness. We call this Nanairo Chlorella, which means 7-colored chlorella in Japanese. These different colors represent different functional components accumulated in microalgae. For example, yellow color represents accumulation of carotenoids, such as lutein. With our unique strains and unique cultivation techniques, we can find a way to produce specific target components from microalgae. With our aforementioned uniqueness, we accelerate our joint project with our industrial partners. For instance, naturally derived color for food application, functional ingredients for nutraceuticals and beauty personal care products, alternative protein for meat alternatives, so on and so forth. So how do we do this? Our joint development project with our partners always start with screening our unique algae strain library to find the best microalgae strain that accumulates target component. Then we optimize our cultivation conditions to maximize productivity of such target component. If necessary, we use our non-GMO breeding technology to mutate microalgae strain. And we can also produce sample products with our pilot scale facility in Japan. These are more specific list of our capabilities. We can provide custom-made joint R&D program to deliver optimized technical package for your needs. We are looking forward to work with companies who are willing to create a new product or new solution derived from algae. If this rings a bell, let's make a movie with us. Let us be your solution for the benefit of not only your company, but ultimately for our planet by cultivating algae's potential for a better future. Thank you.
Hello, everyone. I'm the Sun Xiaojun, the founder and CEO of Bionic M. We are building the powered prosthesis leg for the handicapped people to improve their mobility. First, I'd like to introduce why we are doing this project. In fact, I'm a user of the prosthesis. I got my right leg amputated when I was 9 years old. It was difficult for me to afford a prosthesis. So I worked with a crutch for 15 years. 10 years ago, I got my first prosthesis. It changed my life. After I use a prosthesis or not, I found there are many issues of current products. So I was wondering, could I design better products for myself and other people? I quit my job from Sony and come back to Tokyo University as a PhD student. I have done this research in university for 3 years, and then I got my PhD degree. Right now, I'm a prosthesis user and engineer and the entrepreneur to build a new powered prosthesis for the handicapped people. First, I'd like to introduce the many issues in current products. People, especially the user, got 3 top challenges in their daily life. For example, it's very difficult for them to go up or downstairs. There are big physical burden on their feet and their body. It's very easy to fall down, so they always take care, prevent how to fall down. Besides, there are over 10 million amputees all over the world. In fact, every 30 seconds, people lost their leg because of some diabetes. This is the prosthesis market overview. It's a very strong leading market, and there's no top manufacturer in Asia, including Japan and China. Most of the current prosthesis is passive, so the powered prosthesis is a blue ocean. I'd like to introduce the difference between the passive and powered prosthesis. The passive prosthesis is like the human's leg with only bone and joint, without muscle. The prosthesis has muscle to provide power to help user walk much more easily. So this is the pain point of the current prosthesis. As I said, it's very difficult to go up or downstairs. It's very easy to fall down, and there's a big burden on their body. So we are using the robotic technology to build powered prostheses to improve handicapped mobility. This is a big difference between the traditional product and our product. As you can see the video in left, it's very easy for user to stand up from seat more naturally and more easily. And besides, our procedure can be used to prevent the falling down, which often happen in passive procedures. Besides, as you can see, the user have to go upstairs by step. In our processes, in right video, the user can go upstairs in realistic way, like everyone. We started this research in 2015. We have done this research for 6 years. Finally, we will release our product at the end of this year. This is our business development. We are doing market and sales activities in Japan and China. We will sell this product in Japan and China because it's a non-medical device. In the future, we'll go to US and Europe markets. In Europe, US, it's the medical device class 2. This is our professional team. We have a team in Japan and China. We are doing this project in international. This is our funding situation. We have raised the money from the VC 2 times. We are looking for the next fundraising. This is some awards we got in past. This is our mission. We are trying to power mobility for all the people. That's all. Thank you so much.
Hello everyone, I'm Armando Tikitain from Komoto University, Japan. Komoto University is a national university located in southwest Japan in Kyushu Island. We have about 10,000 students and 5% are from overseas. We have former prime ministers of Japan and celebrated writers in the list of our prominent alumni and professors. Komoto University, being a top global university and research university, plans to act as a driving force for globalization in Kumamoto and strive to take our place in the world as a center for research that also contributes to the community. Through research, we respond to the challenges and issues related to the society and environment. As we all know, we are experiencing these societal and environmental issues: ocean acidification, eutrophication, melting of the ice caps, and so on. These are all related to global warming as a result of the use of fossil fuels. To address these issues, nations all over the world have adapted policies such as Carbon Neutral 2050 and SDGs 2030. Most of these policies addresses global warming with the use of renewable resources like biomass. In the utilization of biomass, conventional heating and catalysts are commonly used. Conventional heating techniques rely on thermal conductivity, where heat is transferred first from source to vessel, and then from vessel to solution. This is a slow and inefficient method of heat transfer, which also results to heat losses, thereby increasing the operating costs. Metal-based and homogeneous catalysts are also being used, but these are hazardous, harmful to the environment, and difficult to separate from the products. Our solution is the synergy of microwave and carbon-based catalyst. Our technology is sustainable. It utilizes clean microwave energy and carbon materials that can also be derived from natural resources. Green, no other toxic reagents are utilized, consequently eradicating the generation of wastewater for neutralization and hazardous byproducts. Energy efficient, because microwave eradicates barriers to energy transfer as heating originates from the system, thereby skipping the induction stage in conventional systems and accelerates the reaction. Environment friendly, pre and post treatments are unnecessary, and carbon catalysts can be easily separated via ordinary filtration. Our strength is on the process development and tailoring of catalysts for a specific reaction or application. Our collaborators will benefit from our approach because microwave apparatus is readily available and easy to commercialize. A small laboratory scale apparatus can be multiplied to reach the same throughput with higher efficiency, avoiding rigors inherent in the conventional scaling up procedures. For example, in a 100 ml reactor, fatty acids can be converted completely to esters in 3 minutes using this technique. Over 60% of cellulose can also be converted to more easily fermentable glucose in only 1 hour. It can also be easily integrated in existing chemical production facilities like a refinery. The technology, being compact, can be easily implemented even in remote community areas for biofuel production, thus also contributing to regional revitalization. This is highly suitable in ASEAN region, which is home to almost 1/3 of the world's biomass potential. We are exploring the application of our technologies to the utilization of abundant biomass resources in ASEAN region. Specifically, we are focusing on algae and agricultural residues, such as oil palm residues. We seek collaborators from various industrial sectors, such as those working on renewable energy, pharmaceuticals, nutraceuticals, and so on, that are interested in our sustainable green approach. We seek strong and sustainable partnerships to contribute to the realization of carbon neutral 2050 and attainment of sustainable development goals by 2030 and beyond. Thank you for your time, and I look forward to working with you in the near future.
This is Professor Shirai from Kyushu Institute of Technology, QTech, Japan. My university and University Putra Malaysia, UPM, have been collaborated since 1994, more than 25 years. We have studied about palm biomass utilization for any environmentally friendly materials. Cellulose nanofiber, CNF, is one of our products. This production method is developed in Satlab's project since 2013 to 2017, sponsored by Japan Science and Technology Agency, JST, Japan International Cooperation Agency, JICA, and Ministry Higher Education, Malaysia. And Professor Hidaya has successfully developed an effective production method to produce CNF and set up her own spin-off company, Zweb Nano. Now, JST kindly supports our project to successfully provide CNF as a sustainable business through the Access program. Okay, I will introduce Professor Hidaya. Professor Hidaya, please.
Thank you, Professor Shirai. Hi, I'm Prof. Hidaya from University Putra, Malaysia. Our product, nanocellulose, is a versatile nano additive mainly because it can be used in multiple applications. Nanocellulose can be used by various industries, for example, in paper industry to strengthen the paper and paper boards, furniture and composite products, car interior, and even in medical, healthcare, and food products, either as a carrier or thickening agent. As a nanomaterial, our nanocellulose has the advantages of being lightweight, renewable, non-toxic, and has high strength. Imagine having a car dashboard which is lighter, but at the same time has the same strength or even stronger compared to the conventional material. Nanocellulose is indeed a superior additive. The current trend, which prefers for the use of sustainable and lighter material, has made nanocellulose as an attractive nano additive. So as seen here, nanocellulose has been used globally. and there is an increased demand for the material year by year. Nevertheless, the producers are mainly from the developed countries. It would be good if this advanced material can be produced in our region, since we are very rich in biodiversity and would have ample local bioresources to produce the nanocellulose. So to realize this, our technology has actually been taken up by a startup company called Zoeb Nano. Zoeb Nano is a startup nurtured by our own university, University Putra Malaysia. Through this startup company, our nanocellulose has been successfully certified as a genuine nanomaterial, which is a certification program by NanoVerify to confirm the quality of a nanomaterial. Another interesting point about our product is that the nanocellulose is produced from agricultural residue and it is a non-wood resource, making the process more sustainable. The advantages of our nanocellulose lie on these three main concerns. The first one is we are the regional producer for nanocellulose, in which this may ensure price stability, just-in-time delivery, and consistent supply. Secondly, CNF is a renewable, biodegradable, and biocompatible material. And thirdly, our CNF is produced mainly from oil palm biomass, making it more sustainable since it is a non-wood resource. Well, we look forward for collaboration with the upstream businesses, for example, raw materials producer in terms of pulp maker, as well as the downstream businesses, for example, the users. The users could be from paper, automotive, composite, paints and coatings, polymer, or even glove product industries. We welcome any form of collaborations, even business partnership and co-investment. Do contact us or visit us at our booth for further discussion. Thank you.
Hello everyone, I'm Shinya Miyagishima from National Institute of Genetics in Japan. So today I'd like to introduce our high-density cultivation of genetically tractable microalgae. So our laboratory in the institute is supported by Japan Science and Technology Agency to develop hot spring microalgae for industrial use. In particular, we are developing unicellular algae cyanidares, which dominate in sulfuric hot springs worldwide. So we found that these microalgae contain higher concentration of proteins, vitamins C, E, and K than other commercially utilized microalgae. So currently, some species of microalgae are cultivated in a large scale, either in closed bioreactors or open ponds to produce supplements for humans, food colorant, cosmetic ingredient, and aquatic feed ingredient. However, these cultures, especially those in open ponds, are easily contaminated by other undesirable microorganisms such as algal predators and toxic organisms. Also, the culture density is relatively low. That's why the production cost is relatively high. In addition, some microalgae are surrounded by rigid cell wall and require huge amounts of electricity to mechanically disrupt the cells to extract the contents. Moreover, no procedure for genetic manipulation has been developed in these commercially utilized microalgae. So to overcome these issues, we have been developing hot spring microalgae cyanidares, such as cyanidium and gardiria. The naturally occurring forms of these algae are surrounded by rigid cell wall, but we have succeeded in generating cell wall-less forms of these algae without any genetic modification. This cell wall-less form also can be cultivated stably. The cell wall-less form can be easily disrupted to extract the cellular contents and well digested by shrimps and fishes. Also, we have developed procedures for genetic manipulation of these cell wall-less new algae. In addition, these new microalgae can be cultivated outside, both in seawater and freshwater-based acidic media. So even in organic medium, The combination of acidic condition and salinity of seawater can effectively inhibit contamination of other undesirable microalgae such as fungus and bacteria. The yield is more than 10 grams per liter, which is more than 50 times of other microalgae in open ponds. So this system largely saves costs for algal production. So we welcome licensing and collaborations, for example, using these microalgae for protein and vitamin source, functional feeds and foods, and cosmetic ingredient. So please visit our booth for discussion. Thank you very much.
Hi, my name is Hiroshi Kawamoto, Executive Director of Reversal and Professor of Kyoto University. Today I'm going to introduce our technology to produce killer T cells from iPS cells to be used as off-the-shelf T cells for cell therapy targeting cancer or viral infection. So then firstly, what are killer T cells and iPS cells? So killer T cells can find the target cells by using the T cell receptor and kill them. So something like this, very strong, potent cells. And iPS cells are stem cells that can give rise to any kind of cells, including T cells, originally invented by Nobel Prize Shin Yamanaka. So Reversal produce killer T cells from iPS cells to be transfused to the patient of cancer or viral infection. So Reversal came from reverse and cell, originally founded 2 years ago in Kyoto City. So this is my own artwork, so trying to express a company to produce T cells in a factory. Okay, so in currently ongoing adaptive T cell therapy, T cells are taken out from patient and transduced with TCR or chimeric antigen receptor gene and then expanded and given back to the patient. So this strategy is really effective for some kind of tumor, but so there remain some issues, time consuming and costly and unstable in terms of quality because T cells depend on patient-derived T cells. Then to solve these issues, we use universal iPS cells as material cells so that regenerated cells will not be rejected by patient. And then cells are transduced and T cells are mass-produced and cryopreserved to be used as off-the-shelf T cells. So it will be universal and rapid, and we can achieve low cost because of scale merit, and we can guarantee high quality homogeneously. And then these are key technologies of our company. So one is a basic method for the production of material cells in which iPS cells are transduced with TCR gene. So we can use any TCR and high quality iPS cells. And then, so the other one is method to induce killer T cells. So we can easily mass produce high quality killer T cells. And this is our actual plan of clinical application. So to plan to realize in 3 years. We plan to treat acute myeloid leukemia patients by targeting WT1 antigen and plan to use semi-universal iPS cells and WT1 TCR gene, so that had been already clinically tested in collaboration with Kyoto University Hospital. And this is, so lastly, I'd like to propose a model of collaboration. I think we can license out our technology. So actually, we have already concluded the research license contract with Otsuka Pharmaceutical Otsuka is now conducting a clinical trial of CAR-T and TCR-T in autologous setting in Japan. But now Otsuka wants to use our method for the development of universal T cells in exclusive and global manner. But this is restricted to a small number of target antigens. So we can license out our technology for other target antigens. For example, our cells can be used as a platform cells so to produce CAR T cells. And also, if you have coronavirus specific TCR gene, then so you can produce T cells that can be used for the treatment of COVID-19. So, okay. Thank you very much for your attention.
I'm Toru Sato of Sonia Therapeutics. I introduce our company and technology from now on. Our company name, Sonia, combines sonics and future. We would like to bring new future to cancer patients using our sonic technology. Sonia was established last year and still new company, but its technology and know-how to develop HIVE therapy system was generated by Tokyo Women's Medical University, Tohoku University, and Tokyo Medical University over the years. This is an overview of US-guided HIVE therapy system Sonia develops. This system is using two ultrasound. One is to see organ and tumor. The other one is to treat cancer. The energy of ultrasound is not so high. Therefore, the system collects multiple ultrasound in one part and heat coagulate cancer tissue. The benefit of HIVE treatment is increasing the possibility of curing cancer without surgery, conversion therapy, repeated treatment because of no exposure to radiation, and dose reduction of anti-cancer drug. We would like to say here that HIVE therapy can be performed for every stage patients because the treatment is minimally invasive. This study was reported by our team members. 4 pet dogs were enrolled and treated by Sonia prototype Moonshot 1. The objective of this study was to certify safety and confirm the validity of MS1. This study could demonstrate safety and efficacy of MS1 HIVE treatment. And this is one of cases. He had end-stage right hip chondrosarcoma. He couldn't put his right hind leg because of pain, like this. However, he could walk by himself 7 days after receiving HIVE treatment. There was no adverse event and the tumor size reduction was confirmed. As a result, he could avoid amputation. Based on basic researches, Moonshot 2 was produced by Tokyo Women's Medical University, Tohoku University, Hitachi, and Denso. This system secures a field of view even two ultrasounds are irradiated at the same time. Some parts were defined based on MS1 experience. Using MS2, clinical trial for intractable cancer was conducted in 2017. This is a video during MS2 HIVE treatment. 11 pancreatic cancer and 1 biliary tract cancer were enrolled. 75% tumor necrosis was confirmed. Along with this, disease control rate was 66%. The percentage is higher than standard of care. There was no grade 3 and 4 adverse event in this study. Remarkably, patients were treated without anesthesia and the treatment time was only 22 minutes in average. We think that this treatment can provide benefit for patients. So we are planning to conduct pivotal study next year. As a next topic, I would like to talk about exploratory research using HIVE. This study is reported by Stanford University, and it shows a possibility of HIVE enhancement of drug permeability. We also conducted basic research using mouse, and we are seeking the possibility of synergy of drug and HIVE. Therefore, if you are interested in exploratory research using HIVE, we are willing to talk about collaborative research with you. This is summary. I'm looking forward to having discussion with you through tech innovation.
Thank you very much for your interesting in our research outcomes. My name is Kiguchi from the University of Tokyo, Japan. So the actually the Professor Oki is leading this project. So today I'd like to introduce our outcomes and expecting the collaboration with you. So our project name is Integrated Water Resources Management with WISE Reservoir Operation. So we called WISE project, supported by the eAsia Joint Research Program. Okay, so firstly, I'd like to explain about the organization profile. So the WISE project is one of the eAsia Joint Research Program during the 2021 to 2023. So the purpose of this project is to co-development of WISE reservoir operation model using prediction of inflow and water demand. And the goal of this project is contributing to the social implementation. So the one which is the dynamic management of dam storage and water quality improvement. So the researcher is in the field of the hydrology, agriculture engineering, and the sociology from the Japan, Thailand, and Laos area. So that we realize the problem statement in the monsoon Asian countries. So recently, due to the climate change, the water-related disasters increasing. So the prediction of the inflow to the reservoir and the estimation of water demand is very important. So far, because it is difficult to operate the reservoir model using the prediction of inflow and water demand estimation with the low availability of information. So the first one, we try to, second one is a green infrastructure for the sustainable water environment. So this is a case study in Vientiane in Laos PDR. So the population growth is very adverse effect to water environment, but because of lacking of the technique and the knowledge, so it's faced to difficulty to keep the water quality. So I'd like to show the technology offers. The first one is the integrated reservoir operation model, so that we try to improve the prediction of the inflow and also the estimation of the water demand using the socio-economic scenario. So the purpose is to develop the high visibility reservoir operation model for multi-space and time scale, like Japan, Thailand, and Laos. And the second one is a green infrastructure for sustainable water environment. It's a dilution and purification by paddy field drainage. We collaborate with a private company in Japan already, but we can collaborate you with these techniques. So the last at last, I'd like to explain about this slide. So of course, research and the collaboration is welcome. And now we are applying to some dams in Japan, Thailand, and Laos using the dam operation model. And we can collaborate with you to contribute some goals of IPCC, SDGs, and the Sendai framework, and so on. So if you are interested in more, please contact with me. So thank you very much.
Thank you for having me today. My name is Harris Vu. I'm a sales representative here at Zenoma in Japan. Zenoma is a startup company spun off from a research group at the University of Tokyo that focused on stretchable electronics. And from that foundation, we created our core technologies, X-Wire and X-Connect, which allow us to seamlessly integrate sensors and devices into clothing to make stretchable, durable, machine-washable smart apparel, which we call Eskin. Eskin's brand goal is to make everyone's lives happier and healthier by making it easier to naturally collect data throughout the wearer's day. Smart devices such as smartwatches are not comfortable for all users, and especially for those with conditions such as dementia, research found they would take off other smart devices. But in a collaborative study we did in Germany, dementia patients were able to wear Eskin for long periods without any discomfort or problems. I'll be introducing two Eskin products today, and the first is Eskin Sleep and Lounge. Eskin Sleep and Lounge uses, like many of our products, a unit we call the hub, which contains a Bluetooth transmitter and battery to power the Eskin after attaching it onto the apparel. This particular hub attaches using magnets and can be easily attached and taken off by anyone, and once taken off, the Eskin apparel is completely machine washable. Eskin Sleep and Lounge can be used by end users and consumers in their own private homes and is especially aimed towards those who live away from their family and allows them to stay in touch through our application. Eskin Sleep and Lounge measures temperature within the clothing as well as movement and movement during sleep to give a sleep score, and also has fall alert detection so that it can alert a registered family member in the case of an emergency. Eskin Sleep and Lounge can also be used in larger settings, such as nursing homes and elderly care facilities, to track and monitor large numbers of residents without the use of privacy-invading cameras. Speaking of cameraless, that brings me to our next product, Eskin Mevo Let's Walk, which is a complete lower body motion capture system that uses 7 IMU sensors embedded in stretchable, comfortable apparel to capture unrestrained human movement. Our particular application uses data from a 10-meter walking test to output 5 gait parameters in an easy-to-understand visual report for the end user. But the hardware is powerful enough to capture the full range of gait parameters comparable to optical motion capture systems such as Vicon and OptiTrack. Eskin Mevo Let's Walk can be used not only in hospital research settings, but can be used for events to gather data from many participants in a short amount of time, such as when we tested it at an event at a major station in Tokyo in September of last year. Over 300 participants took part over 5 days, and we were able to measure up to 10 people an hour. Each participant received their own walking health check report to take home, which we hope can prompt those concerned with their score to get a more comprehensive check from a medical professional. The system itself can output data useful for biomechanics researchers and researchers working in a hospital or university as well. Zenoma is looking for both distributors and customers to help spread our products into markets where we know our technology is much needed. We offer our Eskin planning, development, design, and mass production to all B2B distributors and customers looking to gather raw data in order to create solutions and services using healthcare big data. That brings me to the end of our presentation. Please visit our booth or email me if you'd like to learn more. Thank you for your time.